Thanks very much, Adrian, for this kind introduction. Actually, it has been a great pleasure to be here and listen to the talks, especially since coming from the kind of applied clinical field as a pediatric oncologist. I always find it really helpful to have these interactions with the diagnosticians, and since you can tell from my clinical role, I'm usually a receiver of diagnostic information rather than a generator of diagnostic information. And I can tell you that one of the most puzzling things is getting a 3-page report, which has a lot of details in descriptions, and then in the end it says, highly pleomorphic, slightly anaplastic, highly atypical, mesenchymal tumor, NOS. And now you're stuck as a clinician to do something with this information and to decide about how to treat the patient. And that basically is driving my motivation in my hobby that Adrian was referring to in the lab, to really try and push some of these new technologies more towards clinical application rather than sitting in our ivory tower and say, well, this is all really cool and we can publish high impact papers out of this, but in the end it doesn't really help my patient. So I just wanted to give you some of the introductions into what we are doing and how we are tackling it, and was going to split it into two main parts. One of them is utilizing DNA methylation patterns for tumor classification, and I will also give you some explanation why we think that this might be useful and reasonable. And in the second part, I just want to give you a very brief overview and insight into our clinical cancer genomics project, which is spanning all pediatric tumors across the different entities, and is mostly focusing on patients at relapse because usually they don't have any kind of established treatment option left, and therefore we propose to do kind of a very thorough genomic profiling on them, and just tell you a little bit about our experience with that so far. So as you know, and many of you will be working with these kind of books, almost every cancer entity or group of cancer entities is summarized in these kind of WHO books, and as you can see from the customer reviews, the latest update of the WHO classification for brain tumors is really doing well, apart from the fact that I was surprised that within 2 years we only have 9 reviews, which is slightly puzzling. But anyways, so the question really is on how we can further develop this, and I want to start with a few of the challenges that we are facing, especially in the space of brain tumors, but I'm sure that this is also true for other malignancies. So one of the problems or the challenges that we have for brain tumors is that the proliferation rate as measured by Ki-67 index or similar methods is something that is centrally important in terms of classifying a tumor into low-grade or high-grade category. And in a clinical setting, that can mean that you do nothing but watch and wait and do the most intensive treatment to this patient. And if you take pediatric high-grade gliomas as an example, you basically would expect that in a developing brain, you overall have more proliferation, right, independent of malignancy. And there, the mitotic index is really sometimes very tricky in terms of interpreting what should be called a high-grade versus a low-grade tumor. And if you apply molecular profiling, in this case it's methylation profiling, could also be done by other methods, and then predict in a spectrum of cases that were all called pediatric high-grade glioma, what the molecular features of these would be consistent with, you basically end up with a split like this, where you have the whole spectrum of something that really looks like a high-grade tumor in terms of the clinical behavior, but you also have these cases in gray here, which we would molecularly call low-grade astrocytoma, and they also have the expected outcome of a low-grade astrocytoma. But all of these would be called high-grade glioma, mostly based on the mitotic index that you see down the microscope. Challenge number 2 is that sometimes you have entities, in this case, this is a classic medulloblastoma, which can be a Wnt medulloblastoma, or it could be a group 3 medulloblastoma, both of which are consistent with classic medulloblastoma histology, but the survival is completely different and the biology is completely different. And Adrian has worked in these tumors, so he knows very well. But those challenges where you really have not even a distinction by description in the WHO classification, but you really have the only option to call this a classic medulloblastoma, this would leave you with something that can be biologically and clinically very, very different. And the third big challenge that I wanted to flag is grading. So this is an example where I think some of the most eminent ependymoma pathologists or pediatric neuropathologists in the world were brave enough to publish something in a journal of negative results a few years ago. They basically tried to get to consensus if they call something a grade 2 or grade 3 ependymoma. And this sounds like very close together, but in fact, this means that a patient gets a completely different treatment regimen. And here you can see from these figures here that the best possible pediatric neuropathologists in the world can only agree in 4 out of 5 of them can only agree in about 70% of cases. And if you want to have full consensus, 5 out of 5, it's almost as bad as flipping a coin between grade 2 and grade 3 ependymoma. And if you think about it, this would mean that a patient gets concomitant chemotherapy while on radiotherapy plus 6 months of intensive chemotherapy after radiotherapy. And this makes the difference between a grade 2 and a grade 3 ependymoma. I'm sure that you don't want to be the receiving patients or the receiving doctor who has to make this call because it's really not satisfactory to know how uncertain this is. So what can we do about it? And I think that this whole meeting is essentially praying this, so I don't have to convince you of this. But really, in addition to looking at the scans and looking at the pathology and the morphology, it's really important to come up with a consistent and uniform way of looking at the tumor biology and trying to integrate this with the other data that we can obtain from each patient. So why did we choose to look into methylation patterns? So it all started out with the notion, and everyone knows that obviously, that every cell type in our body obviously has the same underlying nucleic acid code, but the derivative tissues that are really different in their function and in their biology are actually largely dictated by the underlying epigenome. And the easiest part of the epigenome at this stage to assess is the DNA methylation status. So what we think, and can also now prove for one or the other entity, is that actually these DNA methylation fingerprints largely resemble a cell of origin memory than something that undergoes dramatic evolution during tumorigenesis. And this is why we felt that since this DNA-based can be done on paraffin material and has this feature of a cell of origin fingerprint would be ideally suited for tumor classification, assuming that every tumor entity or subtype of a tumor entity is derived from a very specific cell type at a certain time of differentiation of this cell type. And one of the things that we found quite striking when we started this, because everyone has been talking about promoter hypermethylation and downregulation of genes in cancer, is actually when you look at this in a genome-wide fashion and integrate DNA methylation patterns with RNA expression patterns, actually the peak that you get between differentially methylated genes and differentially expressed genes across the genome is actually quite small in the promoter region. So you have relatively few genes that are differentially expressed in a tumor cohort and differentially methylated as an underlying potential mechanism. But the by far higher peak is actually in gene body. Essentially questioning this dogma of promoter hypermethylation in cancer being a driver for these tumors, but rather being a reminiscence of the cell of origin. And the cell of origin actually at some stage was differentiated into a certain tissue type or lineage. And at first, you have a PRC2 signature that is then translated into a DNA methylation pattern that is largely restricted to the promoters. And indeed, these genes are all downregulated and all hypermethylated. But this is not inherent in the tumorigenic process, but rather something that was there in the cell type before the tumor actually started, before the cell underwent malignant transformation. So what we were using at this stage was the 450K and now 850K methylation array of Illumina because of the versatility of this rather than doing whole genome bisulfite sequencing or any enrichment method to really scan the methylome of a tumor. And the big pros, as I already mentioned, are on the one hand that you can use paraffin-embedded material. We also titrated down the input amount of material, and very often we can even get a good result from 25 to 50 nanograms of DNA. We consistently get very good results from 100 nanograms of input DNA. And as the name tells, we are assessing genome-wide 100,000s of different individual CpG sites. The other very strong advantage of utilizing such an array-based method is that from the SNP information that is contained on the array, you can also derive very good resolution copy number plots. So you essentially get 2-in-1 and get this bit for free. We also have published a bioconductor package for the copy number calling from these arrays a while ago, which is accessible. And in this case, you can just see that this is a glioblastoma here with the PDGFR alpha amplification, EGFR amplification, CDKN2A, B deletion, and so on. And most of this is much better in terms of resolution than FISH. So if you have an oligodendroglioma, have a 1P9Q deletion, you basically get hundreds and thousands of individual data points rather than fishing 1 on 1P and 1 on 9Q. So we really, at some stage, our neuropathologists stopped doing FISH analysis after obtaining this information because it's so much more robust. One more thing that we can read out from these arrays is basically MGMT methylation status, which obviously is something that is inherently represented on these arrays, which is very frequently being used as a predictive marker for response to alkylating chemotherapy for brain tumor patients. So when we started all of this, the first thing we did was basically looking into one particular tumor entity and see whether we find meaningful subgroups, such as in this case, medulloblastoma, where we find the 4 different subgroups. Then we looked into high-grade gliomas, where we consistently find the subgroups that are very similar to what is being reported by TCGA, or in ependymoma, where we also find very distinct molecular subgroups. So all of this is nice, but at some stage, you also ask for the clinical applications of this, and consistently throughout this kind of intratumoral questions, we basically got very consistent results in terms of these subgroups being associated with different age and different demographics of patients, with different genetic lesions, with different outcomes, with different response to therapy. So we think that they tend to make a lot of sense. But as a next step, we thought, well, if this works so well, and if you can really group patients into clinically meaningful subgroups that you will not receive by just looking into the morphology, the question at some stage is, if you had a reference cohort that was all-encompassing in terms of all the different brain tumor entities that we can think about, and have one individual patient sample, can you basically attribute this patient sample to one specific class, and what would be the likelihood of a false positive or false negative call in this case? So we set out and generated a very large data set, which consists of about 3,000 individual brain tumor samples, and tried to really span all of the diversity of brain tumors. And this is, at this stage, when we published it, it was spanning about 80 different entities. Now we are approaching 100 different, molecularly different entities that can be readily distinguished only on the basis of the DNA methylation profile. And this already includes some of the subgroups that I just mentioned, such as here the ependymoma subgroups. So you can see that they are still localized closely together in the clustering, but still you can easily distinguish them. However, you might also have situations such as in medulloblastoma, where the Wnt medulloblastomas are located over here and the group 3-4 medulloblastomas are located over here, already indicating how different these are biologically. And then we basically applied a mathematical model, which is called random forest classification, where you basically run 10,000 decision trees for each individual sample across the informative around about 20,000 CpG sites that distinguish between all of these 80 classes that we had defined at this stage. And then for each of these 10,000 runs through the trees, you basically get one decision. And then you can basically come up with a scoring system that of these 10,000 decisions, so and so many % vote for one class. And we basically, and this is arbitrary at this stage, but we basically decided if we get something that is 90% consistent for one individual class of these 80 classes, then we would call it a robust call and report it as a robust classification for this particular tumor. And when we applied this method to about 1,500 prospective cases, actually we saw that in about 3/4 of cases, the result of this would be completely consistent with what the pathologist or neuropathologist has said. In about 12% of cases, we would get a change of diagnosis that is clinically meaningful, meaning that we would treat the patient in a different way. And when we show these results, these discrepant results, to the neuropathologist again, and they would look at the morphology again, only in very few cases, which are the red ones here, that's far below 1%, they would disagree with calling this according to the molecular diagnosis. And in all of these 12% of cases, they would agree with the molecular diagnosis. And then we have another 12% of cases, which is really interesting, which we currently cannot classify. And we now already are, of course, digging into this, and I will give you a few examples on this later. But it's really clear that they are enriched for very rare tumor classes that are happening in 1 in 5,000, 1 in 10,000 of all brain tumor cases. And the other bigger group within these 12% of unclassifiable cases are actually patients with hereditary predisposition syndromes, where the first hit already occurred in the cell of origin. And you can imagine that the methylome of the cell of origin of a hereditary predisposition patient will be different from a sporadic tumor patient. And it's kind of interesting to see that this is really popping out here as different classes. And we are currently trying to build this into the classifier as well, that we can also distinguish between sporadic and hereditary tumors. I cannot tell you for sure that this will unambiguously work, but it looks as if it would be possible to tease them out. Then the reviewers of this manuscript actually asked us to prove that this is also not only possible to do in Heidelberg, but also in other places around the globe, and that this 12% of misdiagnosed cases is really a consistent number that should be put out into the literature for people to work with. And actually, we asked about 5 centers that have been using our classification tool before and had run their own cases, which are actually split between Europe and North America. And it turns out, and this was, of course, nice to see, that across these 5 centers, there is a certain variability in terms of the percentage of discrepant calls. But taking these 400 additional cases from external sources together, we again end up in 12% of cases that get a different diagnosis by applying this method. I just want to give you one entity-specific example, which is quite puzzling, again, from a clinical perspective. We had published a paper about 2 years ago on a series, a worldwide collected series of what was called CNS primitive neuroectodermal tumors. And actually, it turned out that when applying the classification, the methylation-based classification tool to this series that the local pathologist would call CNSP-NET, basically in about 75% of cases, you would get a molecular classification call for an established entity different from CNSP-NET, which could range from high-grade glioma, ETMR, ependymoma, pineal tumors, ATRTs, and so on. And only in about 25 to 30% of cases, you get 4 distinct subgroups by methylation, and all of these are associated with quite specific genetic hits, and all of these are distinguished by methylation, by gene expression, by other measures from all of the other tumor entities that we have looked at. So we think that by kind of cleaning up the cohort first and then try to understand the remaining cases that we could not classify at this stage really helped us to identify 4 new molecular groups of brain tumors, which we now can really follow up on. And they, again, all being lumped together before have quite different outcome data and response to treatment. And now the next step to really apply this further was we have received a cohort of a COG study that was conducted by Jim Olsen out of Seattle, where they basically run a supratentorial PNET study, and again, included all the patients that were locally diagnosed as a CNS PNET. And when basically applying this method to this cohort, and this was done in a completely unbiased and blinded fashion, so we only got the slides sent over to Germany. We didn't know the diagnosis, we didn't look into the morphology or anything like that. And often we even have had to scrape off the material from previously stained IHC slides, because this was the only thing that was left over. And basically in about 75 to 80% of cases of this clinical trial cohort, we were able to work with these previously stained IHC slides. And what it turns out is that the majority of patients who were included on this trial actually had a glioblastoma and not a CNSP-NET. Molecularly. And when you now report back all of these different glioblastoma subgroups to the PI of the study, and they draw the Kaplan-Meier curve having the clinical data, these are the glioblastoma patients in the study, and these are what we would now call CNSP-NET, indicating that all of a sudden CNSP-NET, which we thought was a disease that has almost as bad an outcome as a glioblastoma, turns out to be pretty responsive to chemotherapy, and maybe it's worthwhile keeping these patients on this trial after cleaning up the trial cohort for the contamination of glioblastoma patients. So then we moved on and actually tried to implement this principle in a national prospective clinical diagnostic study, which we call Molecular Neuropathology 2.0, where we combine a methylation-based analysis with a customized gene panel sequencing, which is constituted of 130 genes at the moment. We are now updating this to 170 genes. I'm more than happy to send the gene names to everyone who is interested in running a neuro-oncology gene panel. And what we are doing there is basically taking into account the imaging. So it's central imaging, and the radiologists are looking at this. And whenever we have a discrepant case between the central neuropathology, which is done in a different institution, and our molecular pathology, we basically initiate a weekly telephone call. We are discussing several cases every week now, including the neuroradiologists. Oh, thanks very much. And whenever there is a discrepant case, then we try to get to a consensus after this. And I just want to give you some numbers on this. So one of the numbers that we are pretty excited about is that we get to about 80% population-based recruitment across Germany. So it's across 50 different pediatric oncology centers for all newly diagnosed pediatric brain tumors. One of the things that I always want to stress, and this was a discussion here at this meeting as well, so what do we do with the germline predisposition? Can we really extrapolate this from the somatic mutation calling that we are doing, or do we have to include the blood? I personally believe that we have to include the blood, A, to improve our somatic mutation calls, and B, to also be extremely sure about what we are reporting in the germline. And actually, it turns out that about 12% of this prospective population-based cohort have a clearly pathogenic germline mutation, as evidenced by our human geneticists who have to sign out on this and have to double-check them with an orthogonal method, which then is being reported back to the treating physician, which fundamentally changes treatment approaches for many of these cases, as you know. And I just want to give you one example, which we found quite interesting. So in this early evaluation, which was only 1/3 of the cohort that we have now, we have just enrolled the 1,000th patient last week into this. We basically found a few dicer germline mutations in pineoblastoma, which was expected based on the publication that was out a few years ago. But we also had 2 cases which had one of these kind of descriptive names being called malignant neuroepithelial / malignant sarcomatoid tumor. So you can tell that this was quite difficult. And actually, when now knowing this, we found that there was a whole group of methylation-based individual cases that actually turned out to belong to the same group, and all of them seem to have some sarcomatoid features, and we just published this 2-3 weeks ago, but I think all but one of these cases had a dicer mutation either in the germline or somatically, again indicating that this kind of germline-driven cell of origin probably can lead to a distinct methylation signature here. And another glimpse at the interim data of this prospective study is when we look into what was called high-grade glioma in this pediatric population by reference pathology. We find this kind of puzzling result here that only about 50% of cases, in fact, molecularly turn out to be high-grade gliomas. About 1/4 would be called low-grade glioma, according to the molecular data. And we have this large group of unclassifiable cases, and I will get back to this in the examples of what we find in these little extra islands now, which might explain part of this. But when you recall one of my introductory slides, this is actually what we had expected, but it was still kind of puzzling to the neuropathologists when they said, well, if I call something a high-grade glioma, I want this patient to be treated as a high-grade glioma. But from a clinical perspective, if I can prevent giving radiotherapy and 12 months of temozolomide to a 4-year-old patient with a high-grade glioma or low-grade glioma, I would rather like to spare this patient of this intensive treatment if it turns out to be molecularly a low-grade glioma. And we personally have about 8 or 10 patients during follow-up that were initially diagnosed as a high-grade glioma and unexpectedly survived. And all of these, when retrospectively looking at the methylation data, would basically be called a low-grade glioma by methylation. So particularly for this reason, one of the topics that was chosen for the C impact group, which is kind of a solution of the brain tumor classification group at the WHO for interims updates that are published online rather than in a book, which I think is extremely helpful, was pediatric low-grade glioma, because we really need to speed up here to make sure that we do not overtreat a lot of patients. We also have the first trials starting now utilizing targeted treatments randomized in comparison to standard of care chemotherapy for these patients. So every patient also needs to get a molecular diagnosis to really make sure we can interpret the data of these clinical trials prospectively. I just want to spend a few words on the website that we put up, because we kind of made an experiment with the community to say we offer this classification tool freely available as a web tool, and you can just upload your data from the EPIC array or from the 450K array, and then get a standardized molecular report with a confidence score between 0 and 1 back, and also copy number plot. And then we also asked the submitters whether they would agree and the patient have consented that we can make use of the data that they are uploading for further development of the classifier. And we were really uncertain how this would work out. But as of a few days ago, basically we had, within the first 10 months or so, 14,000 uploads here. And in about 10,000 of these cases, the submitter would agree that we can utilize the data further for the further refinement of the classifier, which we think is also a really cool way of communication and community-based learning, because only by really pulling together these very rare groups of tumors, we will eventually be able to call them something rather than say, well, this is an outlier, we don't really know what it is. We also have started to work this out for sarcomas. And everyone who is involved with sarcoma diagnostics knows that we also have this problem there of these ambiguous cases where we really don't know exactly what it is, and in a clinical setting we have to do something for these patients. We basically have about 2,000 reference cases for sarcomas profiled by now, about 60 different diagnostic categories. And actually, this is just the heat map of the hierarchical clustering of these. And these are the entities that we have covered by now. So it's essentially almost everything that is contained in the WHO classification, apart from the unclassifiable sarcomas or the others and stuff like that. And this classifier is also already, even without this being published at all, is already up on the web, so people can also make use of it. And every single one of these classes also has a short description on the web that basically describes the centrally important genetic features, copy number features, clinical features that we currently are aware of. This would be the DICER1 example here. So I promised you to also give you some kind of an introduction into these little islands that we find now by looking into more data. And actually, we are looking into more than 30,000 individual brain tumor data sets that we have generated on this platform. And now we can really zoom into the islands that were unknown before, or that we've previously called unclassifiable. And I just want to give you a few examples of this. So this was a group where the gray means that we didn't really know what it was. The blue means that it was part of the reference cohort. And in this case, 3 of these cases were called gliofibroma, which again is now, we are talking about 0.1% or less than 0.1% of all of these 30,000 cases. So it's really a very, very small group, and the likelihood of one neuropathologist seeing 2 of these in their entire career is relatively low, indicating that it's not even theoretically possible that the person would recognize the morphological pattern and recall it 30 years later when they see the second. I think that's just fair to say, right? It's none of us will be able to do that, simply. So these cases here really turned out to be interesting because now looking into these 10 or 12 cases that make up this group, all of them turn out to have 2 breakpoints in these 2 genes, and all of them turn out to have a fusion between these 2 genes, which is kind of really nice proof of concept that it makes sense to tease out this group because we have never experienced this fusion in any other brain tumor type before. Second example is a completely unknown group, which was either called PNET, malignant glioma, glioblastoma. I recently had a patient myself, which was called anaplastic oligoastrocytoma, which you already can tell from the name that it was difficult to diagnose it. And consistently, all of these cases have a loss of chromosome 14, which at this stage we don't know what that means, but again indicates that there is a genetic event in this little group of tumors that cluster very tightly together by methylation, and they seem to have some kind of an underlying common genetics. And again, talking about this large cluster, what we called high-grade glioma world here, so to speak, and dig deeper in here. So if you have this little cluster here, which we also had no case, which was very clear in terms of the morphological diagnosis, it turned out that all of these cases either had an amplification of the BLACL1 or the BLACL2 gene, which are known transcription factors, but to my knowledge have never been implicated as oncogenes for any other brain tumor. And we're now looking into the gene expression patterns of these. It turns out that these cases, amongst all of our cases where we have expression data on, also have by far the highest expression of either of these 2 genes, giving first evidence that this will be functionally relevant in terms of tumorigenesis for this subgroup of tumors. So what do all of these examples have in common? And I just want to reiterate this. So we think that we are looking at a cell of origin signature, and since we see specific genetic hits associated with this specific cell type, we think that it's not a coincidence that a certain cell type at a certain time of differentiation is susceptible to specific genetic hits. It also makes sense that this is the case, right? And this is something that we see more and more often now, that we have a very close connection. Once we have really teased out the meaningful methylation patterns of very specific genetic hits being associated with them. So we can probably also turn this around and search the other way around. So I already gave you one example for the DICER1, and I want to give you one more example. So this was a case of a pediatric or infantile glioblastoma diagnosed through our large-scale profiling that we do at Relapse. And actually, this case turned out to have an ALK fusion and was the case within the high-grade gliomas with by far the highest ALK expression. So we were really puzzled because we thought, well, ALK fusions in gliomas, we have never heard about that before. And then we went back to the methylation data, and sure enough, we find a cluster where this particular tumor falls in, where basically about 50 or 60% of cases all have ALK translocations, and we can now firmly say that there is an infant high-grade glioma or infant glioblastoma group that is driven by ALK fusions, which is really interesting also in terms of treating these patients. So with this, I want to summarize the first part and just hope that I could convince you that these DNA methylation fingerprints can serve as a cell of origin signature and therefore be very useful for tumor classification. We think and we are confident that this is a tool that becomes more and more available to different neuropathologists, and we actually have contracted to share the software with about 10 different large sites across the world to really make use of that. And some people felt uncomfortable to use the website for diagnostic purposes because it's not in their control, and especially here in the US, this would be really required to have complete control on the process. So we basically shared the bioinformatics tool rather than the website, so this is working as well. And we really see the clinical utility of this based on the rate of misclassification, which we think is stable around 10–12%. Additionally, we get meaningful molecular subgrouping, copy number profiles from the methylation data. It also works for sarcomas. Many more islands to be discovered and understood. We currently are looking into about 30 of them with different collaborators across the world. Many of these cases have very close correlation between a genetic hit and the defined molecular subgroups. We definitely have the advantage, especially in pediatric tumors, that these genetically less complex diseases are often driven by a single genetic driver or few genetic drivers, so it's a little easier to understand them, really. And with the second part, and I will just skip through this very quickly, I just want to give you another prospect on how we tackle the relapse cases. So this is a program, and it's a population-based program in Germany, which we call Individualized Therapy for Relapsed Malignancies in Childhood. And this is just to remind you of the grim outcome of patients once they experience a relapse after very intensive radiochemotherapy in a pediatric setting. And what we really do here is try to understand the disease biology at the time of the current relapse rather than extrapolating from the primary disease. We have just recently published an article where we also try to infer druggable genetic hits in pediatric diseases, and you can see that there's a huge variability between the different diseases in terms of how often we can find a druggable target for these cases. And what we try to do here is between day 0, which is actually rebiopsy at relapse, until the molecular tumor board, to go through the whole process of doing DNA and RNA extraction, do all the profiling, do the bioinformatics, preparation for the target review board, which basically needs a lot of handy interpretation and data pulling for the clinicians who are not usually always very much into all of the molecular details of the diseases. And this is the recruitment rates here. So we expected to recruit about 1/3 of all relapse patients across the country, probably 50% for solid tumors, less so for leukemias, because leukemias often have a second and third line treatment option, so there's not as much as a pressure. We are also enrolling patients from all of these other countries now. Israel and Poland are about to start. Greece has just started, and we are very closely collaborating with Australia. And also, it turned out to be very feasible to do the frozen shipment between Australia and Europe. So there's not really an argument that this is not feasible anymore. So this is the map as it currently stands. And for these tumors, we really do a very thorough molecular workup, which includes low-coverage whole genome sequencing for copy number calling, whole exome sequencing, RNA sequencing. We still do good old expression arrays, because there we have about 50,000 data sets to compare with, which is much easier than extrapolating RNA sequencing calls and comparing to AFI data. And we obviously do DNA methylation analysis. So low-coverage whole genome sequencing, we get very good copy number resolution, which in our hands is as good as SNP arrays, but cheaper. So we can use the same library that we use for the whole exome later on. We can find this kind of focal high-level amplifications. In this case, it's a neuroblastoma with a CDK4 amplification. We do exome sequencing for obvious reasons, RNA sequencing for fusion detection, also to see whether the mutations that we are detected are actually expressed in the tumor. Gene expression changes, as I said, we really want to understand outlier gene expression. So in this case, it's an FGFR overexpression in a high-grade glioma, and this is the reference cohort for high-grade gliomas. And this is the whole informed cohort, and you can see that this is really outlier gene expression by this definition, because we don't have another case of the high-grade gliomas so far that has a similarly high expression level. So this is what we are already doing. We also look into the germline. We also look into some ADME genes to understand the pharmacogenomics. This is more an exploratory outcome at this stage. It's not something that is reported online back to the treating physician. Now we are also looking into total mutational burden, BRCA mutational signatures, and things like that, where mutational signatures can be predictive of a certain treatment to be applied. And now we are trying to add a few more features to this. We're currently starting a feasibility study on also doing proteomics and phosphoproteomic profiling on them, have started a liquid biopsy program, also try to infer immune infiltration from the RNA sequencing data. So this is more exploratory at this stage. This is what we are basically reporting back to the treating physician. And this is again an interim report, so about 1/3 brain tumors, largest proportion of sarcomas, neuroblastoma, hematological malignancies are underrepresented, and a lot of very rare tumor types where people even at the primary diagnosis don't know what to do, then these patients would also be eligible to be enrolled. So what we try to do systematically from the very beginning is to really not report things based on our gut feeling because we think that this is a good target or could be a target or so, but try to systematically report these features for every single genetic hit that we are finding if there's any evidence for this to be a potential drug target. And this might be the completely wrong approach, but at least it's consistently reported. So the first question, obviously, is this gene or this alteration druggable, yes or no? Then the second question that we are asking, is it a genetic change or is it an overexpression of the candidate gene? Is it a direct or an indirect drug target? What is the evidence level from the literature, from our own or unpublished preclinical data or from clinical data, even better? And is it an entity-specific hit, or is it something that was never described for this entity before? And then we assign a score between very low and very high to each of these individual changes that we are finding. So this will end up in anything between 0 and 10 reported alterations per tumor. We do not take availability of drugs into account, because if we did so for the pediatric population, we would basically end up in almost nothing, as you know. So this is rather meant to be cataloging the frequency of certain genetic mutations to then negotiate with companies about accessibility to drugs, because now for the first time from a population-based study, we can estimate the frequencies, the true frequencies of certain aberrations, and we can estimate how many of these cases we would have in Germany, in Europe, across the world, or transatlantically within 1 year, for example. So this is one of the, or this is their top very high confidence target, and Jackie Beagle already mentioned this yesterday. So this is a case that we got referred to from Israel, where basically we identified an NTRK fusion, and the patient is now put on the NTRK inhibitor, and this was the result after 2.5 weeks, after an unsuccessful chemotherapy where the tumor basically kept growing, which this is one of the things that me as an oncologist really makes much more happy than having another publication on top of what we already have. And even in these very high evidence targets, we also find the first evidence now for these becoming resistant. And again, this is important to understand. So in this case, we basically found that the translocation was not present in the clone that was obviously selected in the next relapse after the treatment on the trial. And this also, and we don't know what this means, but this also had an NF1 mutation, which also might be a resistance mechanism to NTRK inhibition, which we are currently exploring. Again, not surprisingly now after what I've told you before, but one of the things that we're really digging out here is also that in many cases where we, especially for the brain tumors, because we are prepared for that at the moment, where it turns out that what was supposed to be a relapse of medulloblastoma turns out to be a secondary glioblastoma, which probably is radiation induced. And in almost every published medulloblastoma relapse study to date, this was always based, the diagnosis of the relapse was always based on MRIs, and very rarely on rebiopsy at the time of relapse. So if we assume that this number here and this is about 12 cases that we have included now, is any close to reality, that would mean that every relapse medulloblastoma study that was ever published contains a significant number of glioblastomas rather than relapses of the medulloblastoma, which again makes it very hard to interpret this data retrospectively. Again, we looked into the germline, and one of the interesting findings here is that in about 50% of cases, the germline mutation that we find in the patient was not previously known or suspected clinically, indicating that by just applying this unbiased approach, we detect many more of these cases. And the other interesting number here is, because people keep asking that, how many patients or parents would refuse to be informed about the germline data, which we of course have as an option. And I think it's 95% of cases who want to get the results of the germline data so far. So this is kind of not an excuse for the treating physicians who usually don't like to deal with these kind of complicated cases, not to do it for the vast, vast majority of patients. And this is then all being discussed in a molecular tumor board, multidisciplinary, also with the respective clinical trial centers across the country who are responsible for this certain entity. So to have all the experts involved, also with the treating physicians, and very often also the pathologists call in who are interested in this difficult case. And I just want to finish with a few examples here as well. This was called histologically an anaplastic pilocytic astrocytoma, and actually it turned out to have a very high score for low-grade glioma of the posterior fossa. The patient was successfully treated with a MEK inhibitor. This is an interesting case because it also refers to one of the patients that Jackie Beagle mentioned yesterday, where basically this was one of these secondary glioblastomas after medulloblastoma, and it turned out that this patient had a MET fusion in the tumor, which we then, back then, because we didn't have another drug available but crizotinib, treated on crizotinib. And it was quite interesting to see that on the one hand, this quite big tumor completely melted away after 2 months of crizotinib, but on the other hand, kind of at a different section of the MR, and I could show you more of these examples, new tumor manifestations arose that basically obviously did not have the MET fusion, indicating that there we have another problem where tumor heterogeneity obviously is not sufficiently captured in looking into this one individual biopsy that we have done here. And this is a paper out of Michael Taylor's group that is essentially describing this heterogeneity between primary local recurrence and metastatic disease for medulloblastoma patients. And I think that by looking at the data, there was also at least one case included, which to my eyes genetically looks as if it was not a relapse of medulloblastoma, but a high-grade glioma, which basically was thought to be a relapse of medulloblastoma. This is an example here where we found an ALK translocation in a patient with a myofibroblastic sarcoma. As you can tell, this was not operable at this stage. The patient got into an operable stage, then a few years later relapsed with a lung metastasis, which was again sensitive to the ALK inhibitor, which tells that in this case it really was a clonal event that was tumor driving. And the final case, and this is just something to share with you, because I think that this is also something that we have to keep in mind when thinking about how we treat the patients and how we look at relapses. So this was a patient, again, with a glioblastoma with a known V600E mutation, who had an endless therapy with any combinations of temozolomide with something else for several years. And then only 4 years after the primary diagnosis was put on dabrafenib and trametinib in this case, and had a very short duration of a good response and then massive progression. And if you look into the tumor data or in the genetic data here, this was the primary tumor which had a total of 4 somatic mutations, one of them being the V600E mutation. This is the first relapse that we analyzed before the patient was put on trametinib and dabrafenib. There it had 6,000 coding mutations, one of them being the V600E, and you can tell from all the other mutations that the patient has that it's very unlikely that this tumor responds durably to any kind of MAP kinase or any kind of treatment, essentially. So the overlap is 3, including the V600E mutation. So this is raising the question for me whether this patient would not be better off by being treated with a BRAF inhibitor or combination of BRAF and MEK inhibitor upfront rather than first mutating the whole tumor genome and then being surprised that the targeted therapy doesn't work. And to kind of prove that, we also looked into mutational signatures here, and the red signature is the relapse 2, and relapse 3 basically is signature 11, which is thought to be caused by alkylating chemotherapy. So you can now even prove retrospectively that all the other mutations were actually caused by the treatment that we have given to these patients. One thing that we don't understand, or I don't think we understand at the moment, is why does this happen in one patient but not in the other patient? So we always also have many, many patients who were treated with temozolomide for a year or 2 years who basically do not show this massive relapse and this massive accumulation of mutations, and I think at this stage we really don't know. So with this, I want to summarize the second part. I think that it's needless to say that pediatric tumors are fundamentally different from adult tumors. That the etiology is largely unknown apart from hereditary predisposition, which we think is still underestimated based on clinical assessment. We often see these specific oncogenic mechanisms such as fusion oncogenes, single pathway disease, developmental pathways mutated, enhancer hijacking, chromothripsis. That is one specific genetic mechanism or genetic hit that seems to drive tumorigenesis. We definitely can say that relapses are fundamentally different from primary disease and definitely is not appropriate to extrapolate what we give to the patient at relapse from the genetics or the biology of the primary tumor. We can detect druggable alterations in quite a high proportion of patients. Caveat is given for secondary neoplasms as differential diagnosis. And finally, since all of these are really rare, it's really essential to collaborate internationally about these very rare diseases. This I want to stop. I skip this because I'm running out of time. Thank my group at the German Cancer Research Center and the Hope Children's Cancer Center at the NCT. This is the group who is running the molecular neuropathology. It's in very close collaboration with our neuropathologists, Andreas von Daimling's department, and not in opposition to them. This is acknowledging all the people who have contributed to the classifier. As you can see, it's really a worldwide effort. The INFORM team, the other countries that are participating. And with this, I stop and hope that we still have 3 minutes for questions. Thanks very much.